Welcome and thank you so much for coming out tonight for this event. I want to acknowledge a few of the distinguished audience colleagues and collaborators and trainees and family members that are here, and friends who are impacted by Peter's life, both professionally and personally. His wife, Barbara, is up front here, and we have several colleagues who worked in the laboratory with him, including one of the speakers tonight, Ben Wolozin. Peter and I met about 13 years ago when I asked him to join the Charleston Conference on Alzheimer's Disease, which is a conference that we created back then from an organization called New Vision Research, which is the publisher actually of this book. I asked him to join as a senior mentor. It's a conference that we gathered 15 young scientists together, and we've been doing this now for 14 years or so, and we've given away to research funding something in the order of a few $1 million. But it's more than just about the grants. It's about teaching and mentorship and collaboration and getting scientists from different areas of science together, all working on the same problem. One of the things that I noticed when Peter he accepted, obviously, the invitation to come, but he was just a natural teacher and a natural mentor. I mean, he was an amazing individual. He had a way of talking to young scientists that made them feel that they mattered and that they were going to go on and win a Nobel Prize anyway, independent of what they were doing at that moment. And he just gave them confidence in themselves. And one of the things that I noticed in the first conference is I struggled with this, but he somehow found a way to meet and talk to every single one of the young scientists who were invited to come. And that was truly remarkable. And he found one-on-one time where he would sit down with them and ask them, tell me about what you're doing. And just a fantastic teacher and mentor, as I said. Well, tonight we honor Peter's scientific legacy, and we reflect on the human side of scientific discovery. We honor not only what Peter discovered, but the community he helped build. We also gather to celebrate the release of Labs and Labyrinths, the life and work of Alzheimer's research pioneer Dr. Peter Davies, and to continue our conversations about the future of Alzheimer's disease research. If there was any one thing that I could criticize Peter for, is that he was a staunch Yankees fan. And being a Red Sox fan, we had some interesting conversations during the years about who was going to win, and maybe a few bets on the side. But and he was also a fantastic gardener. Just a really, he loved gardening. That's where you'd find him, as you'll read in the book, out in the garden with a Pink Floyd t-shirt on. And as his neighbors knew him, he was a nice neighbor and a gentleman and a gardener who liked music. They had no idea that he was one of the largest researchers as far as his credit of what he contributed to the cause, living right next door to them. So with that, we thought we would dedicate this book tonight by having a lecture series, or a lecture by a couple of the colleagues that he touched. One of which is Ben Wolozin, who's in the audience. He's going to talk to you, and you were his first postdoc. Second graduate student. I got that wrong, didn't I? I got to read the book. Second graduate student. And then Melissa Murray, who's utilized in her research a lot of what Peter discovered and contributed to the field. So enough of me talking, and I'm going to let Ben take over. Ben is from the Department of Pharmacology and Neurology at Boston University.
Okay. I'm Ben Wolozin. I was his second graduate student. So, I kind of I'm here to convey some of the stuff he did during when I was there, but also I think the broader impact to the community and who he was, perhaps not as much as, as a personal person, but as a professional. I came into Peter's lab as a starry-eyed graduate student, where in my eyes he was somewhere between like Sean Connery and Carl Sagan, you think that as a young scientist you choose people because of their, the incredible insight and everything. But I have to admit, when I was in my MD PhD program choosing someone, they were like, someone told me choose by the personality, not what they're doing. And I had never heard of Alzheimer's disease. But someone said, there's this guy, Peter Davies, and he's pretty cool. And so I went up, and there are so many things we did that were kind of kooky. But it started at the very beginning where I like, hi, Pete. I came, hi, Peter, I want to chat with you. He's like, great. And he walks into the cold room and he pulls out a couple beers. And he hands me a beer, and he drinks beer. We sit down at his desk and we just schmooze, talk about science the whole afternoon and drink beer. Now, I'm not much of a beer drinker, but I thought, this is the way to live. This is being a scientist, So I decided to join his lab. So Peter, so different than me. I'm like your classic, my father was a professor in a snooty town I grew up, et cetera, et cetera. Peter is this salt of the earth person who really just didn't have expectations for things. And so I kind of want to give you some of his background. He was, born in 48 to a groundskeeper and a midwife, and he matriculated to Huddersfield College of Technology. Money, doing biochemical research at Port Downs. Now, you might, I know a lot of the stuff underneath, and so it was, making money was hard. So what Peter would do, I think with Ian Simpson, is they would go to bars, they would take Tylenol and file off the label on it, and they would sell them as uppers at bars. That's what he told me. And to make money, and so and so incredible guy. And then he went to matriculate into a PhD program at Leeds, and then did a postdoc. But what you have to understand, the backdrop to Peter, and one reason why his first discoveries were amplified above and beyond simply the finding of, a cholinergic deficit, which was his first thing, was the revolution that was happening in Parkinson's disease. Because in the 1950s, they started experimenting with dopa, looking at L-dopa, and they found that in models kind of Parkinson's disease, that if they gave L-dopa, it could rescue it. And then, have any of you seen the movie Awakenings? So in the 60s, I'm forgetting the neurologist at Einstein did this. Does anyone remember his name? The guy, Oliver Sacks, right. Oliver Sacks, who lived on City Island, drove to work in a motorcycle, crazy guy. But he was one of the first people who gave L-dopa to patients with post-encephalitic Parkinsonism. And as per the movie, for the first time they could move. It was amazing. And so thinking of it, in 1970s, L-dopa was approved, and then carbidopa. So you have, right up to when Peter is making this discovery, his first major discovery, that you have the whole backdrop to society where the discovery of a neurotransmitter leads to a cure and treatment for a major disease. Amazing. So then Peter goes to Edinburgh, and he makes his first major discovery, where he discovers the selective loss of cholinergic neurons in the nucleus basalis of Meinhardt. And so people look at that and they're thinking Parkinson's disease. And in fact, this became and remains a core treatment of Alzheimer's disease. It actually works better for patients with Lewy body disease, but the point is, this discovery led to a therapy. Not quite the amazing impact of dopamine, but still for society, it was that continuum that was fitting in. And so this article just changed people's thoughts about Alzheimer's disease. So it was both an amazing finding and important for society. Robert Katzman saw this and said, we've got to get this guy. And so here you can see Peter way in the background. And Bob Goldman, are you in this picture? I don't know. Anyway, it would be a brown-haired guy. This is Cedric Rain, Su Yan, these people who kind of were important in the field. And so they brought in Peter, and Peter joined Einstein to like do this kind of the treatment of Alzheimer's. And also in this backdrop, in the 70s, the Alzheimer Association was formed, and there was one other important political change, which was that the coding of death was changed from the last thing that killed you, which was usually pneumonia, to the preceding cause, which was senile dementia of the Alzheimer type. And so now you've got all this stuff going together where people are saying, here's a disease, and we're going to solve it. And Peter was right in there in the mix. Lowly little me. I came to Einstein in 81. I had a drink with Peter in 82. And then I started a full-time graduate student in 83. Diana Casper was his first graduate student who he tasked with purifying the enzyme that makes acetylcholine, cholinacetyltransferase. And Peter, they worked so hard on that. Once Peter took a postdoc to a bar and got him drunk on purpose, because it was Bruce Wayner's postdoc. I don't remember the guy's name. Peter came back and he said, ha! I got him drunk, and once he was drunk, he spilled the beans. I've got the method to say, I can drink anyone except an Irishman under the table. But actually what Peter succeeded in doing was removing that postdoc's disinhibition, so the postdoc no longer felt the need to tell the truth, and he fabricated a whole protocol just to get Peter off his back. And so what Peter found didn't work, unfortunately for Diana, but it was hilarious. In 1984, we started immunizing brains. I wanted to immunize with the Alzheimer brain, and someone else would do the control, but Peter said, no, that's too risky. You have to do the control brains. And they started doing the Alzheimer brains, but I really wanted to do the Alzheimer stuff, so I didn't, I'm a competitive little guy, so I told the person doing it, if you throw away any antibodies or anything, just send them my way to look at. And so one of those was ALS50, I remember actually when I found it, walking into the lab with a 96 well plate with one well that was green, that was ALS50, while Peter was talking to reporters about the cholinergic hypothesis. There were always reporters in the lab. And then we presented that, then we had this article in Neuroscience. So So this was an article that kind of set the stage for a lot of Peter's subsequent work, and and it was just striking because you could see all these tangles, and they kind of went around. This is a plaque, and you can see the tangles around it. We really wanted to believe that this was something completely different, like there was a new thing showing up in the brain, and that was like the way Peter thought, like, you know, Carl Sagan or Sean Connery, let's, you know, do it big. And with that, there was, you know, a lot of press. You know, everyone came by the lab, et cetera. You know, Allison, this may still happen to you all the time anyway, but for, you know, the average person, it doesn't. And so, but while all this is happening, our lives are going on. So I, as a young graduate student, didn't realize that Peter was just 10 years older than me. In my mind, he was like this senior, wise person, but he was having his family. So Alwyn and Robert, you've grown up a little bit, thanks to Alwyn for the pictures, but this is my image of them, you know, and I remember we went to his house at a party, and I remember giving him a card where he said you know, when at his 40th birthday, which now seems so young, and we were like, the card was like, the problem with the younger generation is you're not one of them anymore. But there was a big debate about what it bound to, and Peter and I had, like, on leaving the lab, actually, we actually had a massive altercation. And I kind of remember this. We were yelling at each other. I didn't know enough how to prove it, but I had shown a blot showing that ALS50 showed the exact same binding patterns as tau protein, and I was saying this is tau protein, and I wanted to publish that, and Peter correctly, in a way, said, you haven't proven that. You've just proven that they have the same binding pattern. I didn't know about the other techniques at that time. But a year or two after that, it was interesting to actually read over these articles and see the way it happened. There actually were articles showing that ALS50 binds this protein tau, and that it binds kind of a conformation. So what was special about the antibody was that it bound to the conformation of it. And then I think with Bob Bowser and Greg Gisha, Peter went and did this affinity purification, as they called, like they pulled down whatever it was binding to and made new antibodies to it. And that made like a next generation ALS50, which was called MC1. And that turned out to be something that's used even more broadly in the field. But there was still kind of this question about what it binds to. So I want to show this because for those who are scientists, the key element here, done by one of Peter's graduate students, Greg Gisha, an MD, PhD, and Bob Bowser, was here. Bob, raise your hand. Is this here, showing that if you included this and this without this middle part, the microtubule binding domain, and there's Ian Simpson. You could get you could get reactivity, showing that it was a kind of conformational epitope. There are still questions that remain about it. This type of thing was just in the news recently, because it turns out if you look at these same epitopes, the same markers that we associate with Alzheimer's disease, they were present in every one of us when we were neonates. Every one of us. And they're also present in animals when they hibernate. So it's not like, you know, God or whatever created, you know, this pathological change to cause Alzheimer's. It actually has a function It's just that when it reappears in elderly under stress, then you start having aggregation. So all of this is going along, and Peter is just impacting on the field. And in addition, Peter's giving out the antibodies he's made to everyone in the field. We've all talked about that, how much has propelled our research. I'll show you slides about this in a moment. And sorry, I apologize, this looks junky, but this was the only picture I could get, and kind of using Photoshop make it legible. But Peter was given one of the highest awards, the Potemkin Award by the American Academy, of Neurology, and in 2015, recognizing his contribution to the field. Perhaps equally importantly, those of us who were in the lab, we were kind of joking about all the things we couldn't say in the book. But when Peter met Barbara, he calmed down a lot. It's like he had suddenly gotten his space, and life became solid. So Barbara, I didn't interact with you, but I know that he was just much more calm afterwards, and I think you kept him off alcohol and cigarettes and all that, so kudos. You're a special person. And he gave out his antibodies for so many articles. These are just a smattering of them appearing in so many important journals that really propelled the field. And not only basic research, but it also was MC1 was put through for clinical trials. So, just in the last month, they've announced I can't say success, but there are strong hints of success for tau lowering therapies. And the antibodies didn't work, but it comes out of this work. I will say 1981 was 45 years ago. 1982 I came to the lab, 44 years ago. If you had told me 44 years ago that people would now be saying, wow, there's this new therapy based on this new molecule tau, I would say you are joking. That's ridiculous, but that's how long it's taken. And hopefully we'll be going forward with that. But Peter wasn't just about acetylcholine. He wasn't just about the antibodies that he generated. He was a person who was creative, thoughtful, questioned things. The other analogy I have is Richard Feynman. Those of you who know him, know of him, just this person who looked at things and wondered. And so he also did other things. These are just two examples. Resveratrol, promoting clearance of A-beta peptides, and cell cycle reentry. These kind of other ways of looking at the field that Peter worked on. And then he had this, I think with you, Ian, is that right? Ian, I told a story before you got here, and it's apocryphal, and you might know whether it's true, that Peter took Tylenol tabs and saw them. Not true? Okay.
And I have to say that I was here when you said that, and I bet we didn't do that.
Okay.
We did a lot of other things, but
that wasn't it. All right. So there you go. You can take what I say. There's truth. And Peter's impact, like so many important, powerful papers that were done using his antibodies and based on his discoveries of acetylcholine. And then, of course, Melissa Murray is going to talk about, academic legacy. And this doesn't convey the depth of what Peter did, but he has all these people in the field that are kind of go down, consider him their great-great-grandparent. in terms of legacy. So that's kind of the story of how this debonair, handsome young Welshman became this esteemed elder statesman scientist that I think we all adored. Thank you.
That was great to hear those stories. Next, we're going to hear from Melissa Murray, who's at the Translational Neuropathology at Mayo Clinic in Jacksonville, Florida. Melissa.
Right. Well, it is an absolute pleasure to be here. I feel like I spent half of my science life trying to meet Peter. Hopefully the other half will make him proud. He was actually my science grandfather. So he was mentored by Dennis Dixon. I'm going to show you some photos through time of really getting to know the terrific generosity of Peter Davies. So as I thought about this, I could not help myself. But we talk about upon the shoulder of giants we stand, right? It gives us this ability to see further than we could have ever imagined. And it gives us this hope that all that came before us is going to allow us to accelerate this propulsion. And I just like to contextualize, because before I even met him, he had an impact. And I've had the honor of contributing significantly to science over the years, and one of which was to think about how Alzheimer's disease affects us, where in the brain it affects us. And those patterns helped me to actually go from being a scientist to impacting the clinic. And the ability to do that was really looking under the microscope. And in the field that I study, neuropathology, when we look under the microscope, it's not very often funded well. And so the generosity of his sharing really enabled so many of these ideas to catalyze. And so I took my science from under the microscope and really tried to bring it to the clinic. And one of the things that I love the most is to try to help clinicians, radiologists understand what they're looking at when they're looking at neuroimaging or they're looking at biomarkers, now that we have some plasma and capabilities. And once again, without Peter's generosity of antibodies, many of these findings would not have been had. And while this is an interesting interpretation, we have now started drug discovery in the lab, and he has also contributed to that, and hopefully will be a part of the contribution of really helping the individuals that are affected by the tau protein. So we saw this image, and it is iconic. So the Einstein group is this group of individuals where neuropathology just was a mecca. People came from all over the world, and they had these shared ideals. And we saw Peter in the background. I knew Peter through his antibodies. I knew them through these little words and letters, CP13 and PHF1. And my mentor, Dennis Dixon, always spoke very fondly of him. And that fondness was both his humor and then also his provision and kindness. At the time, Su Wei Yan was there, also my scientific grandmother. And through her antibodies, I really was able to expand ideas and some of the hypotheses. And another legend was Bob Terry. And across these little images that you guys see, they're not only beautiful and depict some of the changes that people deal with when they're suffering through Alzheimer's disease, but that brings the brain back to life. And people often think that brain donation, you're just studying the end stage of a disease, but there's so many time points in a disease that helps us to make a difference, helps us to understand those changes. And so Peter's generosity was not just of antibody provision, but of mentorship. He had a way about him, and my mentor, Dennis Dixon, was able to really grow in his scientific knowledge. So he was an MD, but really wanted that scientific, the research. He wanted to be able to ask the questions that nobody had asked. And he was brought up quite well, also with a sense of humor, but really with a sense of science. Peter gave to brain banks, so this is where we will house brain tissue that people will donate. He gave us the antibodies, knowledge, we would bounce ideas off, but he also enabled quite an amazing program that we have in this country, which is the Alzheimer's Disease Research Centers. And these are centers where a group of clinicians and scientists come together with a really specific mission, whatever that version therein is. But many of us in the neuropathology course would not have been able to have as much fun without Peter's generosity. We wouldn't have been able to look down the microscope and ask the questions of why these changes robbed our loved ones. And he enabled that through these beautiful provision of supplies. I was scientifically raised in a brain bank, so I'm a PhD, I'm a scientist, but I was raised by all MDs. And this gave me the ability to speak the language, but also to think about what matters to the people who are treating our patients, and what matters to the people who are trying to understand the changes. And as part of a brain bank, Dennis Dixon will go through and he'll review the changes that the individual had. We provide little counts, and fortunately, pennies to the dollar, Bob Terry had come up with this beautiful method that allowed us to look at it. But we needed more specificity. There are many different types of diseases with this protein that he studied that we wouldn't have been able to gain the insight. And so by his provision of these, we're able to expand not just the ideas about them, but to find the little nuances and the different changes that the people understood. And so when I was in grad school, I was moonlighting in Alzheimer's disease. I was studying normal aging, but Alzheimer's was really my passion. So moonlighting means nights and weekends. And there was an opportunity to collaborate with another group, but I wouldn't have been able to. Fortunately, we had Peter's antibodies, and we had come up with methods where I could scan the microscope slides and then create computer methods to be able to count the changes. And when you can measure something, you can study it so much better. And by measuring under the microscope, we were able to bring it back to the brain, bring it back to the life of the individual. That really gave us that opportunity to see deeper and further. In parallel to this, and finally as I came out of my moonlighting, one of my biggest contributions to science was to understand that people experience the disease differently. So there are individuals that have a very prominent memory disorder. But then the opposite can happen, where there are individuals where the majority of the pathology is on the outside part of their brain. So they stop being able to perhaps reach for something if they have a bit of a visual impairment. Maybe they have some behavioral changes. And in this instance, we actually weren't using his antibodies, but because of the study I just showed you, the unfunded one, I realized that tau was more than just this little sliver in time, that the brain was trying to give us the clues. The brain was trying to show us that these changes that can occur might help us to understand why people experience changes. And what was really exciting is I also studied the cholinergic system that Ben did such a great job explaining. And we found that individuals that were younger, less often thought to be Alzheimer's, actually had a much more greatly affected region and might benefit from the therapeutics that they weren't being offered because they didn't think they were Alzheimer's. And so his reach, even at this point, I still had not gotten to meet him, was already quite far, and helped us to then inform differences not just in age. Individuals that were younger and older experienced the disease differently, males and females, and other types of risk factors that really helped us to sink this in. But I think one of the more important parts was to understand how quickly. Some people experience the disease and it goes by faster than you would have ever wanted, while others can actually live with the disease, missing their keys for a while. And that's a bit different. We need to know if our patients are declining quickly, a little more slowly, or quite slowly, so that we can prepare for different changes and also have those really upfront questions. But I got an invitation of a lifetime, and that was to the Charleston Conference on Alzheimer's disease. And so those contributions of thinking about Alzheimer's a bit different, I thought I finally had my chance to meet Peter. And this year he had some difficulties, so he wasn't able to attend, but he was actually still with me. And so as I was presenting my work, and the idea was to take these different types of Alzheimer's and to be able to better understand how to find these individuals before they get to me under the microscope. So I was kind of in the clouds. I like the big picture. I like to think about the brain in a large level, and then also grounded in neuropathology, looking under the microscope and asking. But Joe Halpern, one of the most selfless individuals you guys will ever meet, him and Charlie Durango actually challenged me to go much deeper than I think my brain ever thought was possible. And I'll talk about that in a minute. But if you get a chance to talk to Joe, he had the opportunity to probably fund his research into next few lifetimes. And yet through his selflessness, he was able to demonstrate to a philanthropic group the plight of the early career researcher. And I have been fortunate in science. I might work 6 days a week and 10 or 12 hours days, but it's these opportunities that made all the difference. And so while I didn't get to meet Peter here, he was very much a part of the studies, because guess what? we used his antibodies and didn't have to pay for them, which made it a lot easier to be able to push forward. And so did Su Yen. And without getting too much into the science, once again, if I can measure something, I love patterns. I can find out some of the answers. And in this instance, we wanted to understand why certain people had differences in the much more involved memory center and less. And in fact, we found an antibody, or we found a protein that looked a lot like Peter's antibodies, but it was actually the first tau binding part protein that we had discovered in the lab that helped us to dig in, once again, much deeper than would have ever been expected. And maybe that's part of the legacy, is that Peter gave us this gift. He gave us these shoulders to walk upon, but he also kind of gave us that inspiration to move forward. I finally did actually have a chance to meet him before this meeting, but one of my favorite moments of Peter was when we were able to invite him to the human amyloid imaging meeting. And so amyloid beta is the other protein that individuals will study. There's 2 different ones in Alzheimer's disease. And human amyloid imaging is very open, right? Tau is an amyloid. It just means that it's folding on itself. But Peter was like, why are you inviting me to an amyloid meeting? And in fact, his plenary, he gets up in front of the whole conference. He wouldn't even say it. Thank you for inviting me to the human imaging meeting. And the crowd went wild. They were so amused because many of them knew that for the longest time, for you to study Alzheimer's disease, you were forced into studying one form of it. And while that has allowed for us to have major discoveries that kept certain groups, he was what we called a Taoist, there were Baptist and Taoists, would be cut off into a corner. And really, actually him going gave him a chance to see that the world was embracing both of those. And his antibodies, once again, helped us for one of our landmark studies to help the world understand that the tau imaging, so this is where we can see tau now while people are alive, was much more closer to an advanced form. And that helped them to understand when they were seeing these changes, what that can help interpret for the patient. And so then one of the more exciting meetings was Tau 2020. We had our own meeting dedicated to the protein that we all loved. And it was so heartwarming to get to see Peter up there for the opening. And I know that he got to see it happen, and he would have been so proud that all that has come. Recently, I was co-awarded with my mentor the Rainwater Prize, and I would have loved him to have been in the audience, but he was certainly in my presentation. There were pictures and, of course, his antibodies, and he still is with us in many different ways, and not all the ways that we will all each understand. But I am really happy to share with you that they are keeping the dream alive. Jeremy Coppell, his dear friend and colleague, is still working with his lab members to really understand not just the antibodies that we've all grown to love, but they also have some new antibodies. But Peter was their eyes. And so I have the absolute pleasure to trying to live up to his great potential. And so I'm helping to characterize some of those antibodies to really help that dream continue. And I think I actually found them a funder, so they'll be able to also share very gracefully as Peter did. But if there's any bit of heartwarming news that I would like to share with you, and while I can't go into all of the details, Peter was a part of the very first study where they found the gene that encoded for the protein that he was very much an adoration of fan. And when Lindy Lee Jacobs, who's given me permission to say this out loud, who's actually a mutation carrier, and I'm pointing out to a structure that I can actually now talk about scientifically. I would not have been able to a few years ago. But where her mutation is was part of this discovery. And I am absolutely delighted to share that we've begun some of our own drug discovery aspects. What I'm showing you here is a model, like if you go on the ultra level, you can actually see parts of tau that allows for us to even here where we actually had mapped his PHF1. And so we needed to be able to approach her mutation from a little bit of a different angle, because just to remind you guys, I usually look down the microscope. Drug discovery is not what my lab does, but Peter's antibodies gave us the potential to learn enough, to come close enough to hopefully at least contribute to that. And I'm very grateful to say the future has arrived, and there are many different opportunities out there that we might be able to make a difference. And so we've started doing some of the computational modeling. It actually looks like roller coasters. It's really wild. And I look forward to updating you all on that in the future. I'd like to thank my lab, who also loves Peter and his antibodies, and to you all for caring. Thank you.
So another member of the senior mentors that go to the Charleston conference is Randy Nixon. Randy's been a good friend of mine for a long time, and he was a good friend of Peter's. And asked him to just say a few words at the end here.
Yeah, I just have a few minutes to say a few things about Peter, and just to thank the two speakers here who really epitomized the progress that was made from the early days when we didn't even know what tau was. Everyone knew it was a cytoskeletal protein, but now to hear Melissa, realize all the promise that she showed at the Charleston conference, and to have the success that she's had as now a professor at Mayo, it's really quite inspiring. And what's even more inspiring is that chart you showed of the numbers of people that have been further inspired and are taking on what tau actually does, because we really don't know what tau does inside cells. We know a lot about it, but we still don't have that fundamental knowledge of what it's doing to cause disease. But I think what I'd like to share is just a couple of very brief remembrances of Peter, because I was in the lab, talk about senior mentors. I was in the lab well before Peter made the discovery of the cholinergic deficit in Alzheimer's disease. And we were amazed at that time of a scientist finally being publicized in a national magazine. And this was something that was like the holy grail of a scientist, to be recognized that we were actually doing important things. And he was the one who literally was the first one to be recognized for that. And the cholinergic system is now re-emerging. So all of what he was doing back then is coming back to be important in different ways, which we don't have time to talk about. The other thing that I wanted to say is just everyone who's been at study sections knows how much fun they are now, and I'm being sarcastic. But back in the old days, there were many times that I was on program project study sections and panels, various sorts, with Peter, and so we got to know each other a lot. But what was really quite unique was his, even though he was very quiet and shy and retiring in many ways, he invited the entire study section to go to a Baltimore Orioles game. We were in Maryland at the time, and so after the study section, everyone trekked into Camden Yards, and as you know, he's a big baseball fan, and they probably were playing the Yankees. I think I was still a Red Sox fan at that point. But the other thing that he did related to that was that some of you may not know that the study sections were not allowed to serve coffee during the coffee break. That was something that went on for decades, and then suddenly they said, we can't afford, it looks bad if we serve coffee, free coffee to these poor people that are here for 2 or 3 days reviewing grants. Peter said, I will not have this. I will pay for the coffee and arrange for it to be brought down. And I don't know if anyone else has taken up the tradition of it, but you know, for many years he was responsible for looking out for the other people that were in the study section and giving them some relief during the coffee breaks. This was the kind of person he was, epitomized by the Charleston conference, where he gave up so generously his time to mentor the people who have become so successful, like Melissa. So I hope that the book that you will see chronicles some of the other things and some of the other stories that really amplify on what I've said as examples of his kindness and thoughtfulness to other people. And I think that we will close, and I will thank again everyone for coming to here, and the family for being here. I think hopefully you have an idea of how people revere Peter and the legacy that he's left in terms of both ideas and products. These monoclonal antibodies will live on for well beyond our lifetimes, and so I thank everyone for sharing in this event. Thank you.
Thank you. Thank you, Randy. I actually, listening to Randy and some others speak, I realized that in the audience here are the mentors, the senior mentors for the Charleston what we call the Charleston Conference on Alzheimer's Disease. It's run by a 501c3 called New Vision Research. New Vision Research published the book, and the conference started 14 years ago, collecting 15 young scientists together and putting them in the same hotel for a weekend with some senior mentors. Partly because when you're young and you go to a conference and you see someone like Peter in the field, you don't have the wherewithal to walk up and say, "Hi, I'm Joe Helper, and I'm a graduate student." You wouldn't have the nerve to do that, and so this is really meaningful for people like Peter, who gave of his time to hang out with young scientists and let them know that they're a real person, and they have the same kind of struggles when they were younger that they might be going through now, and it really meant a lot. And but I realized sitting here that in the audience are some of the senior mentors who have given their time like this to young scientists, and I want to recognize them. Melissa actually was invited back. She was one of the first in the invitees to come to the conference as a young scientist, and now she's a full professor. And she was the legacy of the conference is that we invite, as you become senior, you come back to be a mentor, and that's how it continues. And Melissa was actually a mentor several years ago. Randy Nixon is a mentor and has been a mentor since the beginning. Allison Goat is sitting in the audience, and she's been a mentor for many years. Ben Wolozin was a mentor for 70 years. Ailing is sitting in the back, was a guest mentor, a mentor who came back too. So these are people who come back, they're very busy, they have big laboratories, and they have a lot of their own students, but they give of a weekend, a Thursday, Friday, Saturday. It's a nice weekend in Charleston, but they do that out of the kindness of their heart and really to promote young scientists. So I want to thank Ben and Melissa for coming out, coming from New York and from Boston and Florida, traveling to give their talks. They really gave some character for us to think about with regard to Peter and not only his science, but him as a person. I also want to thank the organizers of the event, Tara Coker in the back and Stephanie Ponte. I get to look like I get the credit, but they do all the work, and they were the ones, you can imagine, we've never published a book before. So we started from scratch, like, what do we do? I mean, how do we get a writer, and who do we publish it with? And all of that was worked out by them. I specifically want to also thank Peter's family for coming. And not only that, his family embraced us when we contacted them. I'm sure you were wondering, what the heck are they doing? What kind of book is this going to be? Is this going to have Tylenol in it, or little questions about that? But we had to reassure them that it was a book to talk about Peter's legacy in the field and what a great scientist and person that he was. And their cooperation, the book wouldn't have been possible without your cooperation. I also want to take a special thank you to Carol Pittleman of the Litwin family and the Litwin Foundation. They are the ones that underwrite the Charleston Conference on Alzheimer's Disease that they've done for years. And they're the ones that underwrote the cost of publishing this book, which we won't reveal, but was significant. I invited Carol to come tonight, but she doesn't make public appearances. And if you notice, the Litwin group, the Litwin family, is the Litwin of the Litwin-Zucker Center that Peter was in charge of. That's the only thing that they have their name on. They don't put their name on anything else. And I can tell you that they're one of the largest philanthropists in New York City. But they don't want the credit and publicly like that. But I want to specifically thank Carol for that. And finally, I want to encourage you to buy a book. And all of the proceeds goes back into New Vision Research and is used to support young scientists. We have our next conference coming up at the end of February 2027, and we do this every year in Charleston. And then every third year, we started a reunion conference, which is a conference in which all of the alumni who have come to the conference over the years come to one large conference, and that's been very successful. The last point I want to make is that one of the reasons this conference has been so successful is that unlike many conferences, it's a conference that invites scientists from different areas of science. So when you're an imager like I am, I go to a conference, I go to imaging talks because that's what I understand. I wouldn't go sit in a genetics talk. It's a shame, but that's when I was a graduate student, my mentor taught me, no, when you go to a talk, you go to sit in a lecture outside of your field. You'll learn something. And at the time, I didn't really understand it. It was annoying because I had to go listen to someone babble on about science that I had no idea what they were talking about. Well, it turns out that that's really where the great discoveries are made, at the interfaces of disciplines. So in this conference, we invite 15 young scientists that are from different fields, imaging, neuroscience, biochemistry, genetics, etc. But they're all working on the same problem. And that's something different than we normally do, want to get them all in the same room and say, hey, you're all working on the same issue. You need to listen to what your problems are and what your challenges are and things that you've discovered, and start learning each other's terminology and things like that so we can better communicate. And what blossoms out of these conferences is collaboration. collaborations across scientific fields, and that's really important in making progress. So I'll end it here, and thank you so much for coming, and I want to enjoy the, I'm getting pointed to in the back. There's a book in the back that we'd like to, if you have comments or stories or something about Peter you want to write down and sign, we would appreciate it if you at least signed the book on the way out, so we'd have some kind of a record that we could give Barbara and her family of who came to the conference. But if you want to have any anecdotal stories about Peter, that's fine too. And we don't have to leave here for a while, and there's some drink and food in the back, so please enjoy it and enjoy each other's company. Thank you for coming.